folks. Welcome to Anatomy on Demand, Lecture 11B, Part 3, where we are going to discuss signal conduction in the nervous system. So, in order for one neuron to talk to another neuron, that signal needs to travel from the axon hillock down the axon to the axon terminal to the terminal bouton. And that requires action potentials and nerve impulses. And there is a difference between them. I know we have used them pretty interchangeably, but the action potential is that electric polarization, whereas the nerve impulse is the actual movement on them. So in the axon, I tend to use the words interchangeably, but obviously in muscle, I can never use the word nerve impulse. And what I want you to realize is this is going to be different in an unmyelinated axon versus a myelinated axon. So let's start first in the unmyelinated axon, where the process is called continuous conduction. And what this means, if you look at the picture on the top right, there's our normal multipolar neuron, and all along its length, we have all these voltage-gauged ion channels along the entire length. So when the action potential is generated at that trigger zone right at the axon hillock, sodium is then going to enter the axon and diffuse for a short distance, giving us that depolarization, which is then exciting voltage-gauged channels just distal to the action potential, generating new action potential, and that continues along the entire length. And so we're going to have this progressive action potential depolarization followed by repolarization along the entire length, producing this uninterrupted wave of excitation that travels towards the terminal boutons. And this is what we have always talked about previously. And so what I need you to realize is that the action potential itself is not traveling along the axon, rather it's continuously producing a new action potential, and then another new action potential, and then another new action potential. And so it, like your textbook said, it's like dominoes falling, where every domino is the new action potential, and when it generates this with the sodium thing, then it goes towards the next one and generates the new action potential. And so that can be measured with electrodes all along the length. In other words, we are going to have this continuous conduction. Now this is fairly slow at about 2 meters a second, but it's totally interrupted. So what do I mean by this slowness? So in this day and age of COVID-19, and we think about social isolation, let's just say you really had to stand in line at the grocery store and you wanted to keep your 6 feet distance between you. So in a normal person who's about 5'5" to 6 feet tall, 5.5 to 6 feet tall, if you go from the tip of your nose and stick your arm out to the side, as in you are abducting it to the tip of your finger, that's about 3 feet. So if you are standing in line and you could stick your arm out and they could stick your arm out and you are touching each other, then you're not socially isolating. So somebody's aerosols can get on somebody else's. But basically, that is the distance that this could travel in 1 second. So it's self-propagating and it's the same impulse at everyone, so it's non-decremental. That's what happens without myelin. So by contrast, myelinated fibers are different. If we look at their ion channels, at those node of Ranvier's, there are thousands of those voltage-gated ion channels in a micrometer. Okay. Thousands of them. And between them, there's only about 25. So 25 versus power of 100 to 500 times the amount, okay? And so if depolarization happens at a node Ranvier, then we are going to have the travel along the axon underneath the myelin. The myelin's now insulated the axon from the extracellular fluid, so you can't be having sodium fused, and so it's not going to be generating a new action potential. So underneath whatever is creating that myelin sheath, whether it's the oligodendrocyte or the Schwann cell, you're going to have decremental conduction under the sheath, where it's getting weaker and weaker and weaker the further away you get. However, please recall that decremental conduction was fast, okay? So that myelin is sealing the axon, preventing sodium. So sodium is moving within the axon, traveling away decrementally. Excuse me. When it reaches the next node about 1 millimeter away, slightly under 1 millimeter away, it's just strong enough to generate a new action potential. Now, if that node is more than 1 millimeter away, then that sodium is too weak to open that ion channel. And basically, you would have to wait for movement. It's not going to happen. So at the node of rendezvous, because the sodium has moved very fast within the axon, we're going to be able to generate a new action potential. Now we have tons of those sodium gates once again, and the signal will be back to its original strength. And although it's slow, but then it will continue very quickly decrementally underneath the next cell or underneath the myelin sheath of the oligodendrocyte until it hits the next node of rendezvous. So basically, the spike of initiation is only happening at the nodes of rendezvous. After you leave the axon hillock, it's the node of rendezvous, then the next node of rendezvous, then the next node of rendezvous, all the way down until you hit the axon terminals. And so this is called saltatory conduction. Saltatory is a word that means to jump. And so it's as if the action potential signal is jumping from one node to the next node to the next node to the next node. Although in reality, you have to realize the sodium underneath is diffusing underneath, causing the new action potential to jump. So it's not really skipping all the axon between them. It's just a different process is happening in the axon covered by myelin versus in the axon in the node of rendezvous. And so basically what this does is it changes the speed of conduction in a myelinated axon. So as opposed to 5 miles an hour, which is the speed with continuous conduction, it changes the impulse to about 250 miles an hour. Okay. So just to refresh your memory or to recall, action potentials are being generated everywhere there is no myelin covering the axon. And then we have decremental transmission occurring between the nodes. Then we have just enough left when we hit the next node of rendezvous to have another action potential through saltatory conduction. So take a moment, pause, look at this slide, and talk yourself through the processes of both the unmyelinated and myelinated axon, and then rejoin once you're comfortable with this material. So continuing along, talking about the velocity of the axon is totally variable. We already talked before about the larger the diameter of the axon, the faster the impulse, and I've just shown you how myelination dramatically increases the speed. So why is this important? This is what explains the fact that if you would put your hand on a hot cast iron pan without a pot holder, you would almost instantaneously drop it, and in fact you drop it before your brain consciously registers the pain, because the movement and the reflexes are carried on myelinated axons. Those motor neurons are myelinated, whereas the sensory, pain, touch, temperature are carried on unmyelinated axons. So folks, that's the end of this third part. It was short and sweet, but we do have one more part to go on. So make sure you watch all 4 for this lecture.